am Dr. Silvan, Assistant Professor, PG and Research Department of Biochemistry, St.Joseph College of Arts and Science, Autonomous, Cuddalore. I am going to present the topic lysosomes. Name lysosomes is derived from the Latin word lying, means to dissolve or to release or to loosen, and soma means body. The lysosomes was the first discovered Christian D. Dew in year of 1955. These structures are termed as the digestive system of the cell or suicidal bags. It is also called as the police force of the cell and stomach of cell. These are present in animal cells, while in yeast and plants, same role are performed by lytic vacuoles. These are membrane-bound vesicles. They vary greatly in size and shape. Prokaryotes do not have lysosomes, but some hydrolyses have been located outside the lysosomes may originate from the endoplasmic reticulum and Golgi membranes from the process of budding off. Golgi network with endosomes which contain molecules taken up by endocytosis at the plasma membrane. Lysosomal membrane are bounded by a single lipoprotein membrane. The lysosome shows polymorphism in different cell types and even within the single cell. Previously, there are two types of lysosomes, primary and secondary lysosomes. The lysosomes latter are of three types: phagosomes, residual bodies, and autophagic vacuoles. The two types of lysosomes mainly primary lysosomes and secondary lysosomes. The primary lysosomes do not contain particle or membrane for digestion. The secondary lysosomes contain particles or membrane for or three. The lysosomes are three types: phagosome, residual bodies, and autophagic vacuoles. A primary lysosome is formed from endoplasmic reticulum and Golgi complex. The enzymes of the primary lysosomes are synthesized by ribosomes. It can secrete its enzyme outside the cell or store them. The phagosome is formed by phagocytosis and pinocytosis of foreign material. The phagosome associates with a primary lysosome and undergoes fusion to form secondary lysosome. The residual body is the final particle containing indigestible material. It is formed when digestion is incomplete. The autophagic vacuole or autophagosome is a lysosome containing some part of cell in the process of phagocytosis is a cellular process of ingestion in which the plasma membrane engulfs the substances and pinches off to form a particle containing vacuoles. The process of phagocytosis are the first one: chemotaxis and adherence of microbe to phagocyte, ingestion of microbe by phagocyte, formation of the phagosome, fusion of the phagosome with a lysosome to form phagolysosome, digestion of ingested microbe by enzymes, formation of residual body containing indigestible material. Last one, the discharge of waste materials. These are all the major process of phagocytosis takes place in the neck. The lysosomes contain these types of enzymes, namely nucleases, phosphatases, lipases, proteases, glycosidases and sulfatases. The nucleases, which include ribonuclease and deoxyribonuclease, break down polynucleotides into nitrogen bases, phosphates and pentose sugars. Phosphatases are acid phosphate, break down phosphate esters to monophosphates. The lipases are lipids, break down them into fragments. The fourth one, proteases, the proteins break down into the constituent amino acids. Glycosidases, beta galactosidase, beta glucuronidase, alpha mannose. These are break down into polysaccharides to monosaccharides. Sulfatases, it act on sulfate esters, breaking down into. The table shows the important lysosomal enzymes present in the lysosomes, namely proteolytic enzyme, lipolytic enzyme, carbohydrate splitting enzyme, nucleic acid hydrolysing enzymes and other enzymes. Lysosome is essentially a bag containing digestive enzymes. The enzymes may be released within the cell itself or outside the cell. The lysosomes serve several functions. They are extracellular digestion, intracellular digestion, hormone secretion, fertilization, autolysis and chromosomal breaks. Pinocytosis. The pinocytosis is the process of engulfment of fluid particles through the plasma membrane. The next one, autolysis. Autolysis refers to the digestion of own cells by the lysosome. It is also otherwise known as programmed cell death or apoptotic lysis. Autolysis occurs during amphibian metamorphosis, insect metamorphosis, etc. Extracellular digestion. The digestion of material outside the cell is called extracellular digestion. Lysosomes release enzymes outside the cell by exocytosis and bring out digestion. Extracellular digestion takes place during bone erosion process. Next, the chromosomal breaks. Lysosome contain the enzyme deoxyribonuclease, which attacks DNA and causes chromosomal breaks and rearrangement. Thus, release of hydrolases from lysosomes can result in structural abnormalities in chromosomes that can lead to cause disease like cancer. Fifth one, fertilization. During fertilization process, acrosome of sperm head ruptures and releases enzymes on the surface of the egg. These enzymes digest the egg membrane and provide way for the entry of sperm nucleus into the egg. This action also activates the egg for the developmental. Thank you.